going to talk about determining the genetic code and how it was created. So there were 3 scientists involved: Nuremberg, Mattei, and Leader. For the first round, Nuremberg and Mattei. So with the help of specific enzyme, Nuremberg and Mattei created a synthetic RNA molecule that was only made up of 1 nucleotide, uracil. They then used 20 test tubes filled with all different amino acids and placed that synthetic RNA in each. They waited to see which amino acid was able to form protein with that synthetic uracil RNA. They were able to determine with that synthetic RNA uracil that it only reacted and created a protein with one of the amino acids, phenylalanine. They repeated this with each RNA, poly C, poly A, and poly G, but they were only able to figure out amino acids for 3 of them. They determined the amino acids for UUU, AAA, which is adenine, and CCC, which is cytosine. Later, although the system was not able to determine the amino acids for all codons, they were able to narrow it down to just a few. Later, Leader and Nuremberg created a new system using a filter-like system to pull and pick out what to test. It reminded me of a pasta strainer in a way. The big important pieces are left to use, while the small and unuseful pieces go through and are discarded. After creating synthetic mRNA sequences, they put them into amino acid where the important tRNAs would bind onto the mRNA. They were able to filter out those unimportant tRNAs and keep the ones that were bound to the mRNAs, which were known sequences formed synthetically. This allowed them to find out which amino acids were on those tRNAs. Since they knew what mRNA it attached to, they were able to match those amino acids to the mRNA codon. Between the 2 studies and approaches, they were able to find out the many different amino acids that reacted with each codon, and they were able to create the codon chart.